So we know how to break down glucose. We can either use aerobic respiration or lactic acid fermentation. And we also know how to build up a glucose molecule. We can take carbon and then build up new glucose molecules. But again, why do we need to build up new glucose molecules via gluconeogenesis? Say you don't have eaten any carbohydrates. Well, where are your red blood cells going to get the energy? Where is your brain going to, and is your brain going to just exist on just ketone bodies? Well, your body will produce its own carbs. It's inefficient in the long term, but it's a way of actually getting the cells that only prefer glucose, like your red blood cells. It's a way of feeding them. Okay, so we're done with carbs, and let's go on to the other macromolecules. So the next one we're going to talk about, or actually to summarize, catabolism. We have glycogenolysis and glycolysis. So again, breaking apart glycogen, breaking apart glucose. Anabolism, glycogenesis is building up glycogen from glucose. And gluconeogenesis is when you take things like pyruvate, shuffle it around, combine it with other carbon to make new glucose molecules. Now, what about fats and lipids and proteins? Well, this is what I hope to get through in the rest of this lecture. Okay, so then now we're going to talk about glycerol. So glycerol is a 3-carbon molecule, and this is what a cartoon is, not exact structure, but I'm just showing you it for simplicity. So glycerol, sometimes called glycerin or glycerine, they're the same molecule. They're this 3-carbon molecule, same structure. And then you have fatty acids. Now, not all fatty acids have 12 carbons like this fatty acid here, but it's just an example. But fatty acids, they tend to have an even number of carbons. So most of the fatty acids in your body are going to have an even number. And again, there are different types of fatty acids and with different numbers of carbons, but I'm just showing you a typical example. Alright, so there's this storage form of fatty acids called a triglyceride. So again, we did cover this briefly in previous lectures. So what is a triglyceride? Common mistake, people think it's 3 glycerols plus a fatty acid. But the tri actually refers to 3 fatty acids. So 3 fatty acids plus a glycerol. You do chemistry, you metabolize, you do an anabolic reaction that builds up a new triglyceride. And now you have a triglyceride. So this is what happens here. Glycerol plus 3 fatty acids is a triglyceride. Now the opposite reaction, when you take a triglyceride and then break it apart, is called lipolysis. So when you do lipolysis, at least on the chemical level, and sometimes people use it colloquially to refer, like especially if you're into like PT or exercise or food science, they might say like lipolysis is getting rid of fat or burning off white adipose tissue and shrinking it. But in terms of chemistry and metabolism, lipolysis refers to taking a triglyceride and then breaking it apart back into a glycerol and fatty acids. So that's lipolysis on the chemical and metabolic level. Alright, so then let's look at our carbohydrates. Now we're going to talk about glycerol. So glycerol is not a lipid itself. So glycerol is part of a triglyceride, but when it's not part of a triglyceride, it's actually more like a carbohydrate. And why is that? Well, it has 3 carbons, it's polar, and the thing is that glycerol you can get a 3-carbon intermediate. So glycerol can be broken down into energy. This is what I'm getting at here. So it can be shuffled around into 3-carbon intermediate and eventually into pyruvate. And then when it's a pyruvate in the form of pyruvate, it can be metabolized like this. Now the thing about glycerol, it also can be used in anabolic processes. So glycerol can be broken down into energy, but it can also be used in gluconeogenesis. So that's pretty much what I want you to know about glycerol. So it can either be broken down into energy or reused somewhere else in the body. Now, triglycerides, again, when you break it apart, we know what happens to glycerol. You can either metabolize it or use it to build something else. But what about the fatty acids? So fatty acids, again, this is why I make a big deal about carbons, because this will play over and over again in terms of metabolism. So a fatty acid is typically an even number of carbons, and there are different depending on the number of carbons. And double bonds, that's more of like organic chemistry, and when talking about the position, that's more organic chemistry. But what we see here is that different types of fatty acids, they have the different names based on the number of carbons and their type of bonds and arrangements. Again, I don't expect you to know this or actually even memorize this at this point, but notice that carbons are very important overall to a fatty acid's properties. And actually, this is why fatty acids are lipids and hydrophobic. Carbon doesn't really form polar bonds, or strong polar bonds, especially if it's a carbon-carbon bond. That's why they're hydrophobic. Now what we have here, so if this is 12 carbons, this is lauric acid I'm showing you right here. So you also have medium chain fatty acids. This seems, I don't know if it's still as popular, but I know like a lot of people were talking about MCT oil and and it's very hot. It was, at least a year ago, it was very hot in nutrition. So notice we have things like caprylic acid, capric acid. They're compared to having a lot of amino or carbons in their chain. They have fewer carbons in this chain. So we have caprylic acid with 8 carbons over here. So this is what these MCTs are. They're just fatty acids still linked to a glycerol molecule, but the amount of carbons per fatty acids, they're not as long as those longer chain fatty acids with 12 plus carbons.